In the name of Allah, who is the most beneficent and the most merciful biggest, Assalamu alaikum. My name is Muqaddas Khalid. I am from batch 3. Today my presentation topic is Nobel Prize of 2020. The announcement marks the first time the award has gone to two women. There are these two women that win the Nobel Prize of 2020 in chemistry. First one is Jennifer Doudna and the second one is Emmanuelle Charpentier. Win 2020 Nobel Prize in Chemistry. Here my name is Muqaddas Khalid and my teacher are Dr. Hammad Majid. I am from University of Management and Technology. Here is the list of table of contents. First I will discuss here abstract of my assignment, then biography, Introduction of Scientists, Nobel Prize 2020 in Chemistry, Mechanism of Classical CRISPR-Cas9 System, CRISPR View of 2020 Nobel Prize, From the CRISPR Draw to Clinic, Safety and Ethical Considerations, Purpose of Precise Genome Editing Technology, Application of CRISPR in Cancer Research, Application of CRISPR in Cancer Diagnostic, and applications of CRISPR-Cas9 in cancer therapy, and then at last references. Abstract. The 2020 Nobel Prize in Chemistry was awarded to Emmanuelle Charpentier and Jennifer Doudna for the development of the clustered, regularly interspaced short palindromic repeats. Here CRISPR is the abbreviation Clustered, regularly interspaced short palindromic repeats. It is a phenomena in which we edit a DNA. It is a gene editing technology that provides new tools for precise gene editing. It is possible to target any genomic locus virtually using only a complex nuclease protein with short RNA as a site-specific endonuclease. Since cancer is caused by genomic changes in tumor cells, CRISPR-Cas9 can be used in the field of cancer research to edit genomes for exploration of the mechanisms of tumorigenesis and development. In recent years, the CRISPR-Cas9 system has been increasingly used in cancer research and treatment, and remarkable results have been achieved. In this review, we introduce the mechanism and development of the CRISPR-Cas9 based gene editing system. Furthermore, we summarize current applications of this technique for basic research, diagnosis, and therapy of cancer. Moreover, the potential applications of CRISPR-Cas9 in new emerging hotspots of oncology research were discussed, and the challenges and future directions were highlighted. Biography. Emmanuelle Charpentier is a French microbiologist, geneticist, and biochemist. She is a director at the Max Planck Institute for Infection Biology in Berlin, honorary professor at Humboldt University, visiting professor at Umea University, and recipient of an Alexander von Humboldt Professorship. Prior to her current appointments, she worked at several other institutions in Germany, Sweden, and the USA and France. Emmanuelle Charpentier's research on a bacterial immune system laid the foundation for the groundbreaking CRISPR-Cas9 genome engineering technology. She has received numerous prestigious awards and distinctions, and is an elected member of several renowned academies of sciences. She is a co-founder of CRISPR Therapeutics and ERS Genomics. Jennifer Doudna, in full Jennifer Anne Doudna, born February 19, 1964, Washington. American biochemist best known for her discovery with French microbiologist Emmanuelle Charpentier of a molecular tool known as clustered, regularly interspaced short palindromic repeats. The discovery of CRISPR made in 2012 provided the foundation for gene editing, enabling researchers to make specific changes to DNA sequences in a way that was far more efficient and technically simpler than earlier methods. Using the CRISPR-Cas9 system, scientists were able to alter DNA to correct genetic defects in animals and modify DNA sequences in embryonic stem cells, an advance that opened the path to germline, sperm, and egg genome modifications in humans. Doudna and Charpentier shared the 2020 Nobel Prize in Chemistry for their discovery and development of gene editing technologies. Now we will discuss here introduction of Jennifer Doudna and Emmanuelle Charpentier. Jennifer Doudna. Doudna spent much of her youth in Hilo, Hawaii. After earning a degree in chemistry in 1985 from Pomona College in California, she went to Harvard University. There, she worked in the laboratory of English-born American biochemist and dentist Jack W. So won the 2009 Nobel Prize for Physiology or Medicine, and in 1989 completed a PhD in biochemistry. In 1994, following postdoctoral studies at the University of Florida under the direction of American biochemist and molecular biologist Thomas R. Chick, who received a share of the 1989 Nobel Prize for Chemistry. She joined the faculty at Yale University. In 2002, she moved to the University of California, where she served as professor of biochemistry and molecular biology. Early in her career, Doudna worked to reduce the three-dimensional structures of RNA molecules, which provided insights on RNA catalytic activity. She later investigated the control of genetic information by certain small RNAs and became interested in CRISPR. CRISPR is a part of bacterial immune system. It originates with RNA sequence from invading viruses that became incorporated into bacterial genomes. The viral sequences reside as DNA in the spaces between short repeating blocks of bacterial DNA sequences. The next time the virus invades the bacterial cells, the spacer DNA is converted to RNA. The Cas9 enzyme and a second RNA molecule attach to the newly coded RNA, which then seeks out matching strands of viral DNA. When encountered, Cas9 cuts the viral DNA, preventing the virus replication. Doudna and Charpentier found that the guide RNA sequence could be changed to the direct Cas9 to a precise DNA sequence. Their discovery quickly transformed the landscape of genome engineering, creating new opportunities for the treatment of human disease. Genome engineering in humans was an inevitable result of rapid advances in genetic engineering technologies. However, little was known about its safety, and its use to edit human DNA renewed ethical concerns, particularly about whether genetic engineering technologies should be used to modify non-disease traits, such as intelligence. In early 2015, Doudna organized an effort that called for a moratorium on human genome editing. And in April of that year, she and colleagues laid out a framework of immediate actions to safeguard the genomes of human embryos against modification. Despite the precautionary effort, however, in April 2015, Chinese scientists reported having altered human embryo genomes via CRISPR-Cas9. In addition to receiving the Nobel Prize, Doudna received numerous honors and awards for her research, including the Gruber Prize in Genetics 2015 and the Canada Gairdner International Award 2016, both shared with Charpentier. Doudna was an elected member of multiple academies and a Howard Hughes Medical Institute investigator from 1997. Here one thing I'll clear you, the advantage of genome editing. There are three advantages: faster and more accurate diagnosis, more targeted treatments, and third, prevention of genetic disorder. Here we will discuss about the introduction of Emmanuelle Charpentier. Emmanuelle Charpentier grew up near Paris and had diverse interests as a youth. She attended the Paris and Marie Curie University, later part of Sorbonne University, for undergraduate studies, earning a degree in biochemistry in 1992. Her graduate studies were carried out at a Pasteur Institute where she investigated segments of bacterial DNA that move around the genome and transfer drug resistance between cells. In 1995, she completed a doctorate in microbiology and remained at the Pasteur Institute for the next year, working as a postdoctoral researcher. She continued her postdoctoral studies at Rockefeller University in New York. After working as an assistant research scientist at New York University Medical Center, she became a researcher associate at Judge Children's Research Hospital in Memphis and subsequently the Skirball Institute of Biomolecular Medicine in New York. In 2002, Charpentier returned to Europe, taking a research position at the University of Vienna. There she discovered a regulatory RNA molecule that controls virulence factors in Streptococcus pyogenes bacteria. With the help of molecular microbiologist George Vogel at the Max Planck Institute for Infection Biology in Berlin, Charpentier identified small novel RNAs in the S. pyogenes genomes and started investigating the bacteria CRISPR system, which the organism uses as part of its defense against viruses. She discovered that S. pyogenes CRISPR system consists of three components: tracer RNA, trans-activating CRISPR RNA, and Cas9 protein as a far simpler organization than she had anticipated. In 2009, Charpentier continued her investigation of the CRISPR system at Umeå Center for Microbial Research in Sweden. With the assistance of Eliza Delchiva, who have been a student in Charpentier laboratory in Vienna, Charpentier showed how the CRISPR system could cut and modify DNA at specific locations in the genome. In particular, Delchiva provided evidence that trans RNA and CRISPR RNA interact to guide Cas9 to specific DNA sequences. Charpentier, Vogel, and Delchiva reported their discoveries in 2010. The following year, Charpentier met Doudna. The two researchers quickly set to work on a collaboration that culminated in their discovery in 2012 of the mechanism by which Cas9 cleaves DNA. The system subsequently was used with great success to target and modify specific sequences in the genomes of various organisms. In 2013, Charpentier co-founded CRISPR Therapeutics, a company that employed CRISPR methodology for gene therapy in humans, with operations in Cambridge and headquarters in Zug, Switzerland. Charpentier was a member of the company's scientific advisory board in 2015. After a 2-year stint at Hohen University Medical School in Germany, Charpentier moved her laboratory to the Max Planck Institute. Charpentier was recognized with numerous honors and awards, including the Canada Gerneder International Award 2016 and the Kavli Prize in Nanoscience 2018. She was an elected member of the Royal Swedish Academy of Sciences 2015 and the European Academy of Sciences and Arts 2018. Now we will discuss here Nobel Prize 2020 in Chemistry honors CRISPR tool for rewriting the code of life. Emmanuelle Charpentier and Jennifer Doudna have been awarded the 2020 Nobel Prize in Chemistry for their work on CRISPR-Cas9, a method to edit DNA. The two scientists were recognized by the Nobel Committee for their discovery that a microbial immune mechanism can be transformed into a tool that can simply and cheaply edit genomes with high precision. In the 7 decades since the discovery of the DNA double helix, technologies for analyzing, manipulating, and making DNA have enabled considerable advances in biological research. But site-specific modifications of genome remain challenging. Previous genome editing tools such as zinc finger nuclease (ZFN) and transcription activator like effector nuclease (TALEN) highlighted the potential of targeted manipulating of gene, but limited targeting capacity and complicated design hampered the usability of these technologies. CRISPR-Cas9 can be considered a real game changer due to its simplicity and efficiency. CRISPR stands for clustered, regularly interspaced short palindromic repeats and is used by bacteria to fight phage infections. In principle, the CRISPR system enables bacteria to recognize genetic sequence of invaders and target these sequences for destruction using specialized enzymes. These enzymes are called CRISPR-associated proteins (Cas) and include the DNA endonuclease Cas9. In 2011, Charpentier's groups identified a key component of the CRISPR system, an RNA molecule that is involved in recognizing foreign phage sequences in bacteria, so-called trans-activating cRNA tracer RNA. A subsequent collaboration of the Charpentier and Doudna laboratories resulted in the landmark 2012 science paper, which kick-started the era of CRISPR-Cas9 genome editing. The paper showed that a bacterial CRISPR-Cas9 endonuclease is guided by two RNA molecules, forming the tracer RNA cRNA duplex to direct site-specification DNA cleavage. Importantly, it was shown that the dual tracer RNA cRNA can be engineered as a single guide RNA that retains two critical features: a 20 nucleotide sequence that determines the target site and a double-stranded structure that binds to Cas9. This created a simple two-component system in which changing to the 20 nucleotides guide sequence can be used to target CRISPR-Cas9 to virtually any DNA sequence of interest. Gene function can be disrupted by CRISPR-Cas9 mediated induction of double-strand breaks, followed by non-homologous end joining repair or gene replacement by homology-directed repair. Thus, we can now knock out any gene of interest in a wide variety of cells and organisms from all three domains of life. However, the applications of CRISPR technology extend far beyond gene disruption. It also allows for specific changings of individual bases or insertion of sequences encoding fluorescent proteins or epitope tags to study cellular protein functions without overexpression. Another powerful application is the possibility to fine-tune gene function by CRISPR-Cas endonuclease. Dead forms of Cas9 dCas9 can be fused with transcriptional activation or repression effectors to control gene expression. Moreover, CRISPR-Cas9 allows for epigenetic alterations. Methyltransferases and acetyltransferases have been combined with dCas9 to modify gene regulation in a more physiological fashion. CRISPR-Cas9 can be multiplexed to edit multiple genes at the same time, and it has been used for genome-wide high-throughput screenings. The versatility of CRISPR-mediated gene editing make it an ideal tool for physiological research, a field that studies biological systems at all levels of complexity. CRISPR-Cas9 has inspired countless applications in basic sciences, agriculture, and medicine. Despite its tremendous translation potential, it should not be forgotten through that this powerful technology has emanated from curiosity-driven basic research. Remarkably, only 8 years after its inception, clinical trials are underway to take to test whether CRISPR-Cas9 may be used to treat inherited diseases such as beta thalassemia or sickle cell disease. For safe and effective clinical use, genome editing needs to be accurate, efficient, and deliverable to the desired cell or tissues. Scientists around the world are working tirelessly to achieve these goals and to advance the ever-expanding report of gene editing technologies. We are witnessing a biotechnological revolution. Then we will discuss here mechanism of classical CRISPR-Cas9 system. Here we will discuss about its mechanism and we explain with its diagram. Mechanism of classical CRISPR-Cas9 system is a heritable adaptive antiviral immune system of prokaryotes that targets infectious invading viruses and bacteriophage and uses RNA-guided nuclease to cut foreign genetic compound. It contains two compartments, one for Cas9 endonuclease and one for single-stranded guide RNA. The sgRNA directs the Cas9 endonuclease to cleave both DNA strands of the target gene in a sequence-specific manner. DNA cleavage occurs at a sequence 3 base pairs upstream of an NGG protospacer adjacent motif, that is PAM. The genome DNA is repaired by double-strand break DNA DSB repair mechanisms after the cleavage. Therefore, the utilization of the CRISPR-Cas9 gene editing system achieves genome modifications by the introduction of small insertions or deletions in DELs through the relatively error-prone non-homologous end joining. NHEG here represents non-homologous end joinings or the high fidelity homology directed repair, that is represents HDR. Here you can see its diagram very clearly. Here by deletion or by insertion, repair mechanism is continue. One side HDR and second one is NHEG. NHEG represents here non-homologous end joining. HDR represents homology directed repair. At the end, one side gives us insertion or deletion product and second one precise editing. So this diagram explains that single guide RNA directs the Cas9 nuclease to a complementary sequence in the genome where Cas9 will induce a double strand break. Double strand break represents DSB. The target genomic locus must be followed by a 5' NGG 3' motif, protospacer adjacent motif PAM for Cas9 to function. DSB double strand break are repaired by non-homologous end joining or by homology directed repair HDR in the presence of the DNA repair template, which can be exploited to introduce precise genetic modifications or exogenous sequences. Now in this diagram, 3 sections A, B, C. Here you can clearly see that the mechanism of CRISPR-Cas9 system. This is the diagram of dCas9 based gene editing tools. Here CRISPR-A represents fusion of deactivated Cas9, dCas9 with activation domain VP64 induces the expression of gene of interest. Here, in case of section 1A. In section B, CRISPR-I fusion of dCas9 with repressor domain CRAB leads to inhibition of gene of interest. Clear? Now in section C, base editing. Fusion of dCas9 with adenosine deaminase or cytidine deaminase enable introduction of point mutation in the genome. Next point, CRISPR view of 2020 Nobel Prize in Chemistry. In this paragraph, we will see the clearly overview of CRISPR view of all over 2020 Nobel Prize. And this Nobel Prize won by the 2 women. First one is Emmanuelle Charpentier and Jennifer Doudna. A crisper view of 2020 Nobel Prize in Chemistry we will discuss in our part 2 video. Thank you.